Recovery rates for many types of diseases, such as cancer, continue to improve. But have you ever wondered why? Earlier diagnostic testing, as well as novel and more targeted medications, play a significant role in helping decrease mortality rates. In the past, broad-spectrum traditional medicines were used, but more recently, scientists are harnessing the power of cellular machinery to produce therapeutic molecules that target specific conditions. These are known as biologics. So, how are these therapeutic biologics developed and how do they eventually reach patients? It begins with hit identification. Through live cell imaging and high-throughput screening, scientists search through hundreds of thousands of potential biologics to identify molecules with optimal disease-targeting properties. Once a hit molecule is identified, structural changes help to optimize its performance and minimize risk of unintended side effects. Larger quantities are then produced through a process known as cell line development. Cells, such as human embryonic kidney cells or Chinese hamster ovarian cells, for example, are considered to be some of the most efficient and productive biological factories. These different cell lines are transfected with a DNA-encoding plasmid containing the gene sequence of the optimized candidate molecule. They are then grown in specialized media under specific conditions, which can be optimized to provide higher cell growth and molecule yield. Further cellular variables are measured to find the best performing cell line to produce the novel biologic. Once the optimal biologic and cell line is developed, vast quantities are manufactured. And with the right regulatory approvals, the new biological medicine can be administered to patients and aid in their recovery. To find out more, visit sartorius.com.